Octopuses are solitary. They hunt alone. They hide alone. They disappear without warning. They do not gather in crowds. And yet, nearly 2 miles beneath the surface of the Pacific Ocean, that expectation begins to fracture. There, along the slope of an underwater volcano, thousands of octopuses have been found gathered together in one narrow stretch of seafloor, not scattered across miles of terrain, not drifting through open water, pressed against the same rocky incline. The depth measures roughly 3,200 meters below the surface. At that level, the water pressure exceeds 300 times what we feel at sea level. Sunlight has never reached this place. The surrounding ocean hovers just above freezing. There are no forests of kelp, no coral cities, no visible source of food, only stone and black water. And yet, in one patch of fractured rock, the temperature rises by a few degrees. Warm fluid seeps upward from beneath the crust, subtle but measurable. Thousands of pale octopuses cling to those warmed surfaces. Many are motionless. Many are curled around clusters of translucent eggs. They are not feeding. They are not hunting. They are not migrating. They are waiting. Researchers who studied the site confirmed that the females had gathered there to reproduce. Each mother anchors herself to the rock and guards her eggs for months. During that time, she stops eating. When the eggs hatch, she dies. Thousands of animals converging in darkness to reproduce once and never leave. But how do they find this precise strip of warmer stone in a landscape so vast and cold? Why gather in such numbers in a place that guarantees death? And what does this hidden garden reveal about life in the deepest parts of the ocean? Tonight, we descend slowly into a deep sea garden where life gathers at the edge of its own ending. But before we begin, if these gentle journeys bring you a sense of calm, I would appreciate it if you gave this video a like and maybe even subscribed. It helps this little community grow. And if you'd be kind enough, let me know in the comments where in the world are you listening from and what's the time is over there. It's always a joy to see how far these sleepy stories drift across the world. Now take a slow breath, let your shoulders loosen, and together we will travel down to the volcanic slope where thousands of octopuses gather for their final act. Far from any coastline that can be seen from shore, the seafloor begins to rise in a place west of central California. There, beyond the familiar edge of the continental shelf, stands Davidson Seamount, an ancient volcano that never breached the surface. It rests hundreds of kilometers offshore, hidden beneath open Pacific water, its summit still submerged in permanent darkness. Unlike mountains shaped by wind or rain, this one was formed by fire beneath the crust. Lava once poured outward and cooled in layered flows, building a vast structure that lifts itself more than 2,000 meters above the surrounding abyssal plain. Yet even at its highest point, it remains far below the reach of sunlight. Its slopes are long and uneven, carved not by weather, but by slow geological settling and the steady drift of sediment from above. The octopus aggregation lies along one of these sloping flanks, at roughly 3,200 meters below the surface. Here the volcano's side angles gently downward into the deep basin. The terrain is fractured, strewn with pillow basalts and hardened ridges where molten rock once met seawater and froze in rounded forms. Between these outcrops lie narrow ledges and crevices, subtle contours that create small variations in current flow and temperature. For centuries, this mountain remained unknown. The ocean concealed it completely, its presence betrayed only by slight distortions in sonar echoes. It was not until systematic deep-sea mapping expanded in the late 20th century that its full outline began to emerge. Multi-beam sonar systems, mounted on research vessels, sent fans of sound pulses downward. Those pulses struck the seafloor and returned, carrying information about depth and shape. Gradually, a three-dimensional portrait of the submerged volcano took form on computer screens, revealing ridges, valleys, and plateaus that no human eye had witnessed directly. As mapping technology improved, details sharpened. Autonomous surveys traced the contours with increasing precision. What once appeared as a vague swell on nautical charts became a defined geological structure with measurable slopes and distinct terraces. Only after these maps existed did targeted exploration descend to investigate specific regions along its flank, including the narrow band where warmth seeps gently through stone. Seamounts like Davidson are scattered across the global ocean, thousands of them rising from the abyssal plains. Their presence interrupts the otherwise level expanse of deep sediment. This interruption alters water movement in subtle ways. Currents that sweep across the ocean floor are forced upward when they encounter a rising slope. As water ascends, it can carry nutrients from deeper layers towards slightly shallower zones. Particles drifting in the water column may settle differently along elevated terrain, concentrating organic matter along ridges and ledges. Rocky surfaces also provide rare hard substrate in regions otherwise dominated by soft sediment. Many deep-sea organisms require firm attachment points, and a volcanic slope offers fractures, cavities, and stable anchor sites. Over time, these physical features foster pockets of increased biological activity compared to the surrounding plain. The result is not a bustling reef in the shallow sense, but a quiet intensification of life shaped by geology. On the flank of this submerged volcano, geography and gentle geothermal warmth converge. A mountain built by ancient eruptions now hosts a congregation shaped by instinct and time. In the vast Pacific Basin, where flat sediment stretches for miles in every direction, this rise in the seafloor creates a small but meaningful deviation. Within that deviation, the garden found its place. The discovery did not begin with a sighting, but with a descent planned in careful coordinates and quiet anticipation. Researchers from the Monterey Bay Aquarium Research Institute set out to survey the flanks of the offshore volcano using tools designed for depths beyond the reach of divers. Their work formed part of a broader effort to understand seamount ecosystems, to document geology and biology in regions rarely visited except by instruments and code. Lowered from the deck of a research vessel, a remotely operated vehicle slipped beneath the surface and continued downward for hours. Its lights remained off during most of the descent, conserving energy while technicians monitored pressure readings and navigation data in a dim control room above. Fiber optic cables transmitted a constant stream of information between machine and ship, allowing pilots to guide the vehicle with precise adjustments of thrusters and articulated arms. When the vehicle approached the mapped slope, its forward lights brightened, revealing the textured rock predicted by sonar surveys. Cameras capable of capturing high-definition video scanned across ridges and ledges. At first, the scene matched expectation: scattered invertebrates, occasional fish, the quiet stillness typical of such depth. Then the field of view shifted slightly, and pale shapes began to resolve against the darker stone. They appeared in clusters, then in layers, then in numbers that exceeded the frame. The operators paused, adjusting focus and angle. What filled the screen was not a handful of individuals, but a concentration so dense that rock surfaces were nearly obscured. Octopuses pressed close together along the incline, their arms draped over stone, their bodies arranged in repeating forms that extended beyond the limits of the camera's light. Inside the control room, voices lowered. The team reviewed the live feed, cross-checking depth and location. The vehicle moved slowly along the slope, revealing that the aggregation continued for tens of meters, then farther still. Some animals were positioned over egg clutches, others rested nearby. The density challenged prior assumptions about octopus behavior at such depths, where isolation had long been considered the norm. At more than 3,000 meters below the surface, biological activity is often dispersed. Food arrives in sparse pulses from above, and most species conserve energy through solitary habits. Large gatherings were known in shallow coastal waters during certain reproductive events, yet in the deep sea, such patterns had not been documented on this scale. The expectation had been for scattered individuals, not a congregation numbering in the thousands. The remotely operated vehicle maintained its steady glide, documenting the scene from multiple angles. Robotic arms collected small samples of surrounding rock and measured temperature differences in the water immediately above the substrate. Data accumulated in real time, yet interpretation lagged behind observation. The sight was clear, but its meaning required careful thought. As footage continued to stream upward through kilometers of cable, the research team recognized that they were witnessing a reproductive aggregation, unlike any previously recorded in comparable depths. The surprise did not erupt into celebration, it settled into focused attention. Screens glowed softly in the control room while the vehicle traced the contours of the slope, recording image after image of a hidden assembly that had unfolded far below human awareness. In that quiet exchange between machine and ocean floor, a new chapter of deep-sea biology entered view, carried upward through light and signal, then stored for study and reflection. Long before its name was spoken in any laboratory, the animal had already perfected its quiet design. Drifting through cold layers of the Pacific, it carried no markings meant for display, no bright signals, no armor, only a gentle form shaped for endurance in a world where softness survives better than spectacle. Scientists would eventually identify it as Octopus robustus, a species now often called the pearl octopus, a title that echoes its pale sheen and rounded mantle. Its body seems almost translucent when illuminated, as though sculpted from diluted moonlight. Pigment offers little advantage in an environment without sun, so its skin holds only faint coloration, a subtle ivory tone that blends easily with mineral surfaces. Muscles lie beneath that delicate exterior, arranged in flexible layers that allow the animal to fold, compress, and settle into narrow contours of stone. Without rigid skeleton or protective shell, it relies on yielding rather than resisting, letting pressure pass through its tissues instead of bearing against them. The absence of an ink sac sets this species apart from many relatives that inhabit shallower waters. In coastal habitats, a cloud of ink can confuse predators or provide cover for escape. At depth, such a defense would serve little purpose. Water movement is slow, visibility limited, and encounters with visual hunters are rare. Over evolutionary time, the organ diminished, its function unnecessary in a realm where concealment comes from stillness rather than disguise. Energy that might have supported ink production instead nourishes processes more valuable for survival in sparse conditions. Its eyes are modest in size, tuned less for forming images and more for sensing faint shifts in surrounding light. Vision plays a minor role here, while touch and chemical perception guide awareness. Along each arm, rows of suckers sample the environment with quiet precision, tasting traces carried in the water and feeling textures finer than grains of sand. These senses form a map not of shapes and colors, but of gradients and signals, a different kind of landscape assembled through contact and drift. The mantle, rounded and smooth, encloses organs adapted for slow metabolism. In cold depths, life proceeds at an unhurried pace. Heartbeats remain steady and restrained, conserving energy as oxygen moves through blue blood, rich in copper-based hemocyanin, a molecule suited for binding oxygen efficiently in low temperatures. Even motion reflects this economy. Rather than frequent bursts of jet propulsion, the animal often glides with minimal effort, adjusting buoyancy and posture so that movement requires little expenditure. Arms extend with quiet grace, connected by a web of thin membrane near their base. This webbing forms a soft umbrella shape when spread, useful for gathering small prey or shielding delicate eggs. Each limb carries countless nerve cells, granting a distributed awareness that allows the arms to explore while the central brain rests in calm coordination. Intelligence here does not announce itself through speed or display. It resides in subtle decisions, in patient adjustments, in responses so gentle they scarcely disturb the water. All these traits compose a life fitted precisely to its surroundings. The pearl octopus does not conquer its habitat, it harmonizes with it. Pale skin, reduced defenses, pliant muscles, and quiet senses together create a creature that belongs fully to the deep, as if the ocean itself had shaped a thought and given it form. Beneath the volcanic slope, motion continues even in stillness. Rock that appears solid and settled is threaded with passages so fine they cannot be seen, a hidden lattice where seawater drifts slowly through mineral veins. This movement begins far below the seafloor, where residual heat from ancient magma lingers in the crust. The heat is not fierce, not explosive, only a quiet remnant of the forces that once lifted the mountain upward. As seawater seeps down through cracks, it encounters this stored warmth and changes character. The liquid expands slightly, grows lighter, and begins to rise again through neighboring fissures. This circulation forms a gentle convection system within the stone itself. No chimneys tower here, no dark plumes billow into the ocean. Instead, the exchange remains subtle, a slow breathing of the earth that releases warmth in delicate threads. Where these threads reach the surface, the surrounding water grows just a few degrees warmer than the vast cold beyond. The difference is small enough to escape casual notice, yet sensitive instruments register it clearly. Temperature probes lowered near the rock record gradients, so slight they could be mistaken for error if measured elsewhere. Here they persist, steady and localized, forming narrow zones of mild heat along the slope. Such understated geothermal flow differs greatly from the dramatic hydrothermal vents found along mid-ocean ridges. Those systems roar with mineral-rich jets heated to extremes, shaping towers of metal sulfides and supporting dense communities adapted to chemical energy. The seepage along this seamount is quieter and far older, the fading echo of volcanic activity rather than its fiery voice. Heat disperses gently through porous basalt, moderated by distance and time until it reaches the seafloor as a soft exhalation. For developing eggs, even this modest warmth alters the passage of life. Temperature influences biochemical reactions within each embryo, guiding the pace at which cells divide and tissues form. In colder conditions, metabolism slows, stretching development across longer spans. A slight rise accelerates these processes without strain, allowing growth to proceed more efficiently while remaining stable. The effect resembles a clock whose hands move with calm certainty instead of lingering between moments. Within these warmer patches, incubation periods shorten compared with those in surrounding terrain. Eggs exposed to such conditions can complete their formation sooner, reducing the duration during which they remain vulnerable to disturbance or decay. The advantage is quiet but significant. Time itself becomes a resource shaped by geology, measured not in hours or days, but in the steady progression from fertilization to hatching. Minerals carried upward with the warmed water also play a role. Trace elements dissolve from the rock during circulation, then settle lightly over nearby surfaces. These deposits can influence microbial growth, forming thin biofilms that alter local chemistry. Though nearly invisible, such films may help stabilize the immediate environment around developing eggs, buffering subtle shifts in acidity or salinity. The result is a narrow band of conditions more consistent than the broader surroundings. All of this arises without spectacle. Heat travels through stone, water answers with motion, and the seafloor releases a faint warmth into the ocean. The process unfolds continuously, neither beginning nor ending within human timescales. In that quiet exchange between crust and sea, a gentle thermal refuge takes shape, defined not by fire or steam, but by a softness measured in degrees. Silence defines the surrounding plain more than any landscape ever could. Beyond the gentle rise where life gathers, the seafloor stretches outward in vast continuity, a level expanse of fine sediment, laid down grain by grain over ages too long to measure. Each particle settles from distant waters above, drifting slowly until it comes to rest, forming a soft mantle that smooths the Earth's contours into broad, patient horizons. The water here holds a steady chill, lingering just above its freezing point. This temperature rarely shifts, remaining constant across centuries, as though time itself has settled into stillness. Cold at such depth is not sharp or biting, but persistent, an unchanging condition that shapes every movement and every breath taken by the creatures who dwell within it. Chemical reactions slow, muscles contract with care, and growth unfolds at a pace that would seem almost suspended elsewhere. Currents pass through this realm with similar restraint. Rather than rushing, they drift, guided by distant forces from tides and planetary rotation. Their motion is so gradual that suspended particles may travel for days without covering the length of a human hand. These slow flows carry oxygen and faint traces of nutrients, distributing them thinly across the plain. Nothing arrives quickly, and nothing is easily replaced once gone. Food is scarce, appearing mostly as what oceanographers call marine snow. This gentle fall consists of microscopic remains, fragments of organic matter, and the occasional larger piece descending from the living world far above. Each speck represents energy captured in sunlight long ago, now sinking through darkness to nourish life that has never seen the surface. The supply is unpredictable. Some seasons bring slightly richer descent, while others pass with almost nothing at all. Because nourishment is so limited, abundance seldom gathers in one place. Most animals here live alone, spaced across the sediment like distant stars. A sea cucumber may inch along for weeks without encountering another of its kind. A small crustacean may spend its entire life within a territory no wider than a room. Density remains low, not by chance, but by necessity, for the environment cannot sustain large populations clustered together. Sound travels differently as well. With few obstacles and little activity, vibrations move cleanly through the water, yet there is almost nothing to produce them. No crashing waves reach this depth. No storms disturb the bottom. No reef structures crackle with constant motion. The quiet is not emptiness, but equilibrium, a state in which disturbance rarely occurs, and stillness becomes the prevailing condition. Even the chemistry of the water reflects this restraint. Oxygen concentrations remain modest, yet stable, balanced between slow circulation and minimal consumption. Minerals lie dissolved in delicate proportions, neither depleted nor abundant. The entire system functions like a slow-breathing organism, inhaling and exhaling across spans of time that pass unnoticed by creatures with shorter lives. In such a setting, activity tends to be measured and deliberate. Survival favors patience over speed, efficiency over excess. Many inhabitants possess soft bodies, reduced skeletons, or elongated forms that conserve energy. Some may wait motionless for hours, sensing faint signals carried in the current before deciding whether to move at all. Life persists here, not through intensity but through quiet persistence, shaped by conditions that reward restraint and continuity above all else. Reproduction in octopuses begins not with spectacle, but with contact so brief it might pass unnoticed in the immensity of the sea. A male approaches with deliberate calm, extending a specialized arm known as the hectocautilus. Along its length travels a packet of genetic material, carefully transferred into the mantle cavity of the female. The exchange is precise and restrained. Once complete, the male withdraws, his role in the process largely finished. Within the female's body, fertilization occurs internally. Eggs develop gradually inside paired ovaries, each one encased in a protective membrane and enriched with yolk that will sustain the embryo through its earliest stages. This preparation unfolds over weeks, guided by hormonal rhythms that move quietly through her system. As the eggs mature, her behavior begins to shift. Feeding slows. Movement becomes more purposeful, as if guided by an internal current. When the time arrives, she selects a stable surface, often beneath an overhang or along a firm ledge. Using the delicate tips of her arms, she cleans the chosen area with careful strokes, removing sediment and microscopic growth. The rock must be free of debris, smooth enough to support adhesion, secure enough to remain undisturbed. Only then does she begin to lay. Eggs emerge in long, translucent strands, each capsule shaped like a tiny grain of rice and tethered by a fine filament. She attaches them one by one to the stone, arranging them in clusters that hang downward like pale garlands. The process can take hours, sometimes days, depending on the number produced. Thousands may be placed in this way, forming a quiet constellation anchored to the rock. Once laying is complete, her posture changes. She draws her body close to the clutch, arms spreading outward and then folding inward to cradle the mass. The webbing between her limbs forms a living canopy, shielding the eggs from passing currents and wandering scavengers. She remains there, suspended above them, adjusting her position with minute shifts that keep water flowing gently across their surfaces. This flow is essential. Developing embryos require oxygen, and stagnant water would deprive them of it. With subtle pulses of her mantle and slight movements of her arms, she maintains circulation, ensuring that fresh water bathes the eggs continuously. At intervals, she touches them lightly, clearing away particles that settle from above. Each contact is careful, guided by sensitivity refined over countless generations. During this period, she no longer hunts. Her digestive system gradually quiets, and stored energy begins to sustain her. Muscle tissue diminishes slowly as nutrients are redirected toward maintenance and vigilance. The choice to remain is not conscious deliberation, but biological certainty. If she were to abandon the clutch, sediment could smother it, predators might graze upon it, or microbial growth could spread unchecked. Her presence becomes the difference between survival and loss. Time lengthens within this vigil. Days pass into weeks, then into months. The embryos inside each capsule transform from simple clusters of cells into fully formed miniature octopuses, their eyes and arms taking shape within the translucent casing. Throughout it all, the mother stays close, a steady guardian bound to the rhythm unfolding beneath her touch. Time moves differently inside a living body than it does in the world around it. Within soft tissues and quiet organs, invisible signals keep measure, marking phases that no eye can see. In certain creatures, this inner chronometer carries a single destination written from the beginning. Biologists call this pattern simulparity, a life strategy in which an organism reproduces once and then completes its span. The sequence is not accidental or caused by exhaustion alone. It unfolds through precise chemical instructions, as if a hidden script waits patiently until the final chapter is ready to be read. Though it may seem unusual, this rhythm appears across many branches of life. Pacific salmon follow it as they leave the ocean, swim upriver, release their eggs and milt, and gradually fade. Certain species of bamboo flower only once after decades of growth, scattering seeds before their stems wither. Some insects, including mayflies, spend most of their existence maturing unseen, only to emerge briefly, mate, and fall still. Across water, forest, and air, the pattern repeats, suggesting that a single culminating effort can, in some circumstances, serve survival more effectively than many smaller ones. In octopuses, this turning point begins, the moment reproduction concludes. Subtle alterations ripple through the bloodstream as hormone levels change balance. Compounds that once supported appetite and tissue repair decline, while others rise and redirect energy toward remaining tasks. Metabolism adjusts accordingly. Feeding responses weaken, digestion slows, and the body begins to draw upon its own reserves. These shifts do not arrive as illness or injury. They emerge as orderly transitions, guided by molecular messengers moving from organ to organ with quiet authority. At the center of this transformation lies a small structure near the brain known as the optic gland. Despite its modest size, it acts as a master regulator, releasing signals that influence growth, reproduction, and the closing phase of life. Experiments conducted in controlled settings have shown that when this gland is removed, the expected decline can pause. Individuals may resume feeding, regain strength, and live far beyond the span normally observed. Such findings reveal that the process is not simple depletion but regulation, a coordinated sequence initiated by this cluster of cells and carried throughout the body by chemical cues. What appears from the outside as a fading is therefore an expression of biological design. The animal is following instructions inscribed within its physiology long before maturity, instructions shaped across evolutionary time. By channeling all remaining energy into reproduction and care of offspring, the species ensures that resources are not divided between competing demands. The individual yields, so the next generation begins with greater provision. In the stillness of the deep, this quiet conclusion is neither sudden nor chaotic. It is the final movement of a cycle written in chemistry, completed exactly as the inner clock intended. Cells respond as they were shaped to respond, and each signal arrives in measured sequence. Nothing hastens, nothing delays. The pattern holds with gentle certainty, like a tide that turns because it must. Within that certainty, life fulfills its single promise and comes to rest. Deep within the octopus, nestled among folds of neural tissue, lies a structure no larger than a grain of rice, yet entrusted with authority over the entire arc of its life. This structure, known as the optic gland, rests near the brain and communicates through silent chemical signals rather than electrical flashes. It does not think, perceive, or remember. Instead, it regulates. Like a quiet metronome hidden inside living matter, it sets the tempo for growth, maturity, reproduction, and the gradual closing of the body's long composition. For much of the animal's existence, the gland remains restrained in its activity. It releases measured amounts of hormones that coordinate development, ensuring tissues mature in harmony and energy is distributed where needed. These secretions move through the bloodstream, touching distant organs and adjusting their function with subtle precision. Muscles strengthen, nerves refine their sensitivity, and internal systems align with one another, all guided by signals so delicate they leave no visible trace. When reproductive maturity arrives, the gland alters its pattern. Its chemical output shifts, not abruptly, but with deliberate progression. Hormones associated with mating readiness increase, influencing behavior and physiology at once. Feeding rhythms begin to change. Tissue maintenance slows, and priorities within the body are reassigned. The transformation resembles a quiet change of seasons within flesh, a transition directed from a single point, yet felt everywhere at once. Scientific study has revealed how central this gland truly is. In carefully observed laboratory settings, researchers once removed the optic gland from adult individuals shortly after reproduction. What followed surprised them. Instead of declining as expected, the animals resumed ordinary activity. Appetite returned, strength stabilized, and their lives extended far beyond the span typically observed in their species. The experiment demonstrated that the final phase of life is not caused solely by exhaustion or damage, but by biochemical instruction issued from this small organ. Such findings illuminate a principle woven through evolution. Life does not always favor longevity for its own sake. In certain ecological contexts, directing all remaining energy into a single reproductive effort can increase the survival of offspring, even if it shortens the parent's existence. The optic gland serves as the biological switch that enforces this strategy. Once activated, it sustains a cascade of hormonal messages that gradually silence processes unrelated to reproduction and care. From a molecular perspective, this cascade involves steroid-like compounds and peptide signals that influence gene expression in distant tissues. Cells respond by altering how they repair themselves, how they process nutrients, and how they regulate internal balance. The body does not fail suddenly. It follows instructions embedded within its chemistry, instructions refined across countless generations. Each signal is part of a sequence, and each sequence leads inevitably toward completion of the reproductive cycle. The presence of such programming reflects an evolutionary trade-off shaped by environment and survival pressures. Resources and deep habitats can be unpredictable, and producing a single, well-provisioned generation may offer greater continuity than repeated attempts spread thinly across time. By channeling every reserve into one final contribution, the organism ensures that its lineage continues with maximum support. The gland's command, therefore, represents not a malfunction but a culmination, the last directive in a life designed for this precise outcome. Within that small cluster of cells, destiny is translated into chemistry. Signals pass outward, organs respond, and the living system follows the path laid out within it from the beginning. Hunger arrives quietly, not as a sharp pang, but as a gradual thinning of strength. For a brooding octopus, the first days after laying pass with little change, she remains poised above her clutch, attentive and steady, drawing upon stored nutrients accumulated during earlier months of hunting. Those reserves lie within her tissues as lipids and proteins, carefully built in anticipation of this extended vigil. As weeks stretch onward, the balance shifts. Without feeding, the body begins to metabolize its own stores. Fat deposits are broken down first, converted into usable energy that sustains circulation, respiration, and the subtle movements required to tend the eggs. The process is orderly, regulated by hormones already set in motion. Nothing is wasted, nothing rushed. Energy is allocated with precision, directed toward maintaining posture and gentle ventilation over the developing embryos. When lipid reserves diminish, protein catabolism follows. Muscle fibers, once used for propulsion and hunting, gradually yield their substance. Structural proteins are dismantled into amino acids, which enter metabolic pathways to support essential organs. The arms grow thinner. Strength wanes in increments so gradual that the change might be imperceptible from one day to the next. Internally, enzymes work steadily, transforming tissue into fuel. Throughout this period, the digestive system becomes less active. Enzymatic production slows, and the gut shrinks slightly in response to prolonged inactivity. Circulatory rhythms adjust to a lower baseline, conserving oxygen and reducing unnecessary expenditure. The animal's entire physiology narrows its focus to a single priority. Repair processes that once addressed minor cellular damage are reduced. Growth ceases completely. The body enters a state of controlled decline, not chaotic failure, but strategic conservation. Remaining with the eggs imposes this cost because absence would carry greater risk. Unattended clutches are vulnerable to small scavengers that graze upon unguarded capsules. Sediment settling from above can accumulate, blocking oxygen exchange and fostering microbial growth. Even subtle disturbances in current flow may alter temperature or chemistry around the embryos. The mother's presence mitigates these threats. By adjusting her arms and maintaining water movement, she sustains conditions that no inert surface could provide. Metabolic rate continues to decrease as reserves dwindle. Oxygen consumption drops in parallel, reflecting the body's adaptation to prolonged fasting. Organs reduce activity to the minimum required for survival and vigilance. Neural responses slow, though awareness remains sufficient to detect changes in the immediate environment. The transition resembles a dimming light rather than an abrupt extinguishing. Each system yields incrementally, guided by the same internal programming that initiated reproduction. Over months, this controlled expenditure leads toward an irreversible threshold. With energy stores nearly exhausted and muscle mass significantly reduced, the capacity for recovery disappears. Yet the eggs beneath her arms approach full development, nourished by the steady conditions she has maintained. The cost borne by her tissues translates directly into the stability surrounding them. In this exchange, sacrifice is not dramatic or sudden. It unfolds as a measured redistribution of matter and energy from parent to offspring, mediated through time. Her body becomes the final reserve, converted gradually into the continuity of the next generation. By remaining, she increases the probability that the embryos will reach completion under optimal care. The depletion is therefore not merely loss, it is transformation carried out cell by cell until the vigil has fulfilled its purpose. Within each translucent capsule, life advances in increments too small to witness directly. The change is constant yet unhurried, guided by instructions folded into strands of genetic code. At first, the embryo resembles only a faint cluster of dividing cells. Gradually, those cells organize, forming layers that will become arms, mantle, eyes, and delicate internal organs. The sequence unfolds with careful timing, as though each stage waits for the previous one to settle before continuing. Early development proceeds slowly, especially in deep ocean species whose physiology reflects the chill of their surroundings. Cell division depends on enzymes that operate more efficiently at slightly elevated temperatures. Where conditions remain colder, The same reactions occur at a measured pace, stretching the incubation period across many months. During this time, the embryo relies entirely on the yolk, enclosed within its capsule, drawing energy from that reserve while its body takes shape. In areas touched by gentle geothermal warmth, the process gains subtle momentum. Even a difference of a few degrees can influence molecular motion inside living tissue. Proteins fold more readily, membranes assemble with greater ease, and metabolic pathways complete their cycles with less delay. The effect is not hurried growth, but steady refinement. Development remains orderly, yet the interval between one stage and the next shortens, allowing embryos to reach maturity sooner than they would in colder surroundings. This reduction in incubation time carries quiet advantages. The longer an egg remains unhatched, the more opportunities arise for disturbance. Microorganisms may settle upon the capsule surface, grazing in thin films. Small, wandering animals might brush against the cluster. Environmental fluctuations, though slight, can accumulate over extended durations. By shortening the developmental span, warmer conditions reduce exposure to such risks. The embryos complete their formation before many potential threats have time to act. Location also contributes to their survival. Eggs placed along stable rock faces experience fewer shifting sediments than those laid upon open seafloor. Fine particles drifting through the water tend to slide past inclined surfaces rather than settling thickly upon them. This keeps the capsule walls clear, allowing oxygen to diffuse inward without obstruction. Adequate oxygen supports the growing tissues within, sustaining their metabolism until hatching. Predation pressure in these sites remains relatively low. Many animals that feed on eggs prefer accessible clusters in regions where food is abundant. In sparse environments, potential predators are fewer and more widely dispersed. The narrow bands where broods gather therefore function as subtle refuges, not through physical barriers but through the simple absence of frequent visitors. Survival here depends as much on rarity as on defense. As weeks pass into months, distinct forms become visible inside the capsules. Tiny arms curl and uncurl. Eyes darken as pigments develop. The yolk sac diminishes while muscles and nerves strengthen. Eventually, each embryo fills its chamber completely, its movements gentle but purposeful. At the final stage, a thin membrane gives way, and a fully formed juvenile emerges, already capable of independent motion. Higher hatching success rates, observed in these warm zones, reflect the combined influence of temperature, placement, and time. None of these factors acts alone. Together, they create conditions that favor completion of development with minimal interruption. What began as a fragile cluster of cells reaches its conclusion as a living animal prepared to enter the surrounding sea. The transformation is quiet, continuous, and precise, carried forward by the steady interplay between environment and biology. Somewhere in the open Pacific, long before any gathering becomes visible, a signal begins its journey. It has no color, no sound, no shape. It is only chemistry, molecules released into seawater and carried outward through invisible pathways. Diluted again and again as they drift, these traces spread across distances that no animal could measure, yet they remain detectable to senses tuned for precisely such faint information. Octopuses perceive their surroundings largely through touch and taste combined. Along their arms lie receptors capable of identifying dissolved compounds at extremely low concentrations. As an individual moves across the seafloor, these receptors sample the water and the surfaces it contacts, registering gradients too subtle for most other creatures to notice. A slight increase in a particular chemical signature can indicate that another of its kind has passed nearby or that a suitable habitat lies ahead. Currents assist in shaping this guidance. Though movement in deep water is gentle, it is rarely absent. Water masses flow along predictable routes influenced by planetary rotation, underwater terrain, and differences in density. Chemical traces released from one location can be drawn into these slow streams, forming elongated plumes that stretch across the seafloor. An octopus encountering such a plume may follow its faint gradient, adjusting direction incrementally as concentrations strengthen or fade. The contours of the seabed also play a role. Slopes, ridges, and depressions affect how water travels, channeling it along certain paths while diverting it from others. Animals moving close to the bottom can sense these physical changes through contact with the substrate. Texture, incline, and vibration provide orientation, creating a tactile map that complements chemical perception. By combining these cues, an individual can maintain a steady course without any need for distant vision. Instinct contributes another layer of direction. From the moment of hatching, behavioral tendencies are embedded within the nervous system, patterns refined across generations through natural selection. These tendencies influence movement at key stages of life, guiding juveniles toward feeding grounds and later prompting adults to travel when reproductive maturity approaches. The timing of these migrations often aligns with internal physiological cycles, ensuring that departure occurs when the body is ready. When many individuals respond to similar internal rhythms and encounter the same environmental signals, their paths can gradually converge. Each animal follows its own sensory impressions, yet those impressions originate from shared sources in the surrounding ocean. Over time, separate journeys lead toward a common destination, not through coordination, but through parallel response to the same subtle guidance. Despite these insights, the full process remains partly concealed. Tracking the movement of soft-bodied animals across vast depths presents technical challenges, and much of their travel occurs beyond observation. Instruments can record chemical distributions and current patterns, but translating those measurements into lived experience requires inference. The routes taken, the pauses along the way, and the precise cues that trigger final arrival are still being studied with patience. What can be seen suggests a navigation system composed of many quiet influences rather than a single guiding force. Chemistry drifts, water flows, terrain shapes, movement, and instinct responds. Together, they form a network of direction spread through sea and body alike. Within that network, thousands of travelers find their way toward the same narrow stretch of seafloor, arriving one by one until the gathering exists. Stillness becomes a posture long before it becomes a necessity. After arranging her clutch, the female settles into a position shaped by both anatomy and purpose, drawing her body inward until it forms a living enclosure around the delicate capsules. Her arms bend at gentle angles, curving toward one another until their tips nearly meet beneath her. The thin membranes between them stretch softly, creating a sheltered chamber that holds the developing young within a cradle of muscle and skin. This posture is not rigid. It adjusts in slow increments, each movement measured and economical. A slight tightening of one arm changes the flow of water through the cluster. A gradual shift of the mantle alters her balance against the stone. These refinements occur without haste, guided by tactile impressions that travel along the length of her limbs. Through countless sensory cells, she remains aware of each capsule's position, each filament's tension, each faint vibration passing through the surrounding water. Remaining so close requires restraint. Motion diminishes until it is barely perceptible, limited to the subtle actions necessary for maintaining the eggs. Muscles once used for swift propulsion now serve mainly to hold form. The reduction in activity lowers her metabolic demand, allowing stored energy to last longer. Even her heartbeat slows slightly, matching the quiet tempo of her vigil. In this state, patience becomes a physiological condition rather than a decision. Her skin contributes to this stillness through its remarkable adaptability. Specialized pigment cells, known as chromatophores, lie embedded within the outer layers, accompanied by reflective structures and light-scattering tissues. By expanding or contracting these elements, she adjusts her appearance to resemble the surface beneath her. Tones shift toward muted shades, textures align with surrounding contours, and her outline softens until it blends with the stone. The effect is not a dramatic transformation, but a gentle merging, as though her body were an extension of the rock itself. Breathing continues with equal moderation. Water enters the mantle cavity in slow pulses, passing over delicate gill filaments, where oxygen diffuses into the bloodstream. The rhythm is steady and unhurried, calibrated to supply just enough oxygen for her reduced activity. Each intake is followed by a soft release, producing a faint current that brushes across the eggs before drifting away. This ventilation serves two purposes at once, sustaining her own tissues while refreshing the environment surrounding the embryos. Energy conservation extends to nearly every system. Digestive processes remain dormant. Muscle tone stays relaxed except where support is required. Neural activity narrows its focus to immediate sensations, filtering out unnecessary responses. The body behaves like a carefully managed reserve, distributing its remaining resources only where they are essential. Over time, this conservation allows her to maintain position far longer than constant movement ever could. As days accumulate into weeks, the posture itself becomes a form of architecture. Arms act as walls, membranes as canopy, and the mother's body as foundation. Within this living structure, the eggs remain sheltered from disturbances that might otherwise displace them. The arrangement is both protective and efficient, shaped by generations of refinement. In such quiet dedication, form and function merge completely. The curve of each limb, the softness of each breath, and the stillness of each moment contribute to a single purpose held close beneath her. Before any clutch is formed or guarded, another presence passes briefly through the same realm, subtle and easily overlooked. Male octopuses arrive with a different purpose and a different span of time written into their bodies. Their lives tend to be shorter, not through accident or hazard, but through a pattern shaped by physiology. Growth brings them steadily toward maturity, and maturity leads quickly toward reproduction, after which their role in the cycle nears completion. When a male encounters a receptive female, his movements remain measured and deliberate. He extends a specialized arm that carries packets of sperm, transferring them with careful precision. The exchange is quiet, almost ceremonial in its restraint. Once it is complete, he withdraws, leaving the fertilized future within her care. There is no lingering attendance, no shared vigilance. His path continues elsewhere, guided by instincts that do not draw him back. Inside his body, internal chemistry begins to change soon after mating. Hormonal signals shift, redirecting resources away from growth and maintenance. Appetite often diminishes, and metabolic processes gradually alter their priorities. Unlike the female, whose remaining energy is devoted to guarding eggs, the male has no brood to protect. His physiology, therefore, follows a different timetable, one that moves toward conclusion without the prolonged vigil that defines her final phase. This divergence produces a distinct mortality pattern. Females remain visible for extended periods because their brooding keeps them anchored to one place. Males, by contrast, disperse after mating and continue traveling through the seafloor landscape. Their movements may cover wide areas, yet they are seldom encountered again. Some likely perish not long after reproduction, their bodies yielding to the same internal programming that governs the species. Others may persist slightly longer, depending on individual condition and environmental factors, but none enter a stage comparable to brooding. The absence of males near egg clusters is therefore not coincidence. Their biology does not include structures or behaviors suited for extended care. Without the specialized instincts or hormonal shifts that sustain maternal vigilance, remaining beside the eggs would provide little benefit to the offspring. Evolution has shaped each sex for complementary roles, one focused on fertilization, the other on protection and incubation. By dividing these responsibilities, the species allocates its energy efficiently across the reproductive process. Physical differences reflect this division. Males often possess slightly modified arm features related to reproduction, while females develop internal capacity for egg production and storage. These distinctions influence not only anatomy but behavior as well. A male's life emphasizes searching and locating, guided by sensory cues that lead him toward potential mates. A female's later life centers on stillness and attendance once fertilization has occurred. Each path fulfills a separate requirement within the same life cycle. Because males depart and disperse, their presence is rarely documented in the same places where brooding females gather. Observations of large congregations, therefore, show almost exclusively mothers and their clutches, giving the impression of a community composed entirely of one sex. In reality, another chapter has already passed through, completed its purpose, and moved on beyond sight. The males leave little trace, except the genetic contribution carried within the eggs, a quiet legacy drifting forward into the next generation. From within the final thinning of each capsule, a small body presses outward and meets the surrounding sea for the first time. The hatchling emerges fully formed, no larger than a thumbnail, its mantle scarcely wider than a coin. 8 slender arms extend in miniature symmetry, already equipped with functioning suckers and responsive nerves. There is no larval transformation awaiting it, no prolonged dependency. The moment of release marks the beginning of independent life. Its skin holds a faint translucence, allowing internal organs to show as soft shadows beneath the surface. Pigment cells are present but subtle, capable of gentle shifts rather than dramatic displays. The eyes are proportionally large, prepared to register contrast in dim conditions. Each movement is tentative yet complete, as though the animal carries a blueprint of adult behavior scaled down to its small frame. There is no parent beside it once it drifts free. The protective curve that sheltered its development remains behind, and the hatchling enters water that feels vast and cold. Buoyancy and instinct guide its first motions. Some young rise slightly into the water column, carried by slow currents that distribute them across wider areas. Others remain closer to the seafloor, exploring surfaces with cautious contact. In either case, survival depends on immediate responsiveness. The environment into which they emerge offers little forgiveness. Food is not abundant, and predators may be larger relatives or entirely different species sharing the same depths. Small fish, crustaceans, and wandering scavengers can consume a hatchling in a single encounter. Even subtle environmental fluctuations challenge bodies that have only just begun to regulate themselves outside the capsule. Energy reserves inherited from the yolk sac diminish quickly, and the young must locate prey before those reserves are exhausted. Growth during these early stages requires frequent feeding on microscopic or small invertebrate life. The hatchling's arms probe for movement, guided by chemoreceptors and touch. Each successful capture contributes to a narrow margin of survival. Failure carries immediate consequences. Mortality rates during this period are high, not because of weakness in design, but because of the sheer scale of the ocean relative to their size. Vastness alone becomes a hazard. Yet within this apparent fragility lies an evolutionary calculation. By producing thousands of eggs, a female distributes risk across many individuals. Even if only a small fraction reach maturity, that fraction can sustain the population. This strategy reflects the balance between energy invested in each offspring and the unpredictability of their surroundings. Instead of concentrating resources into a few heavily provisioned young, the species opts for numerous well-formed but self-reliant hatchlings, each equipped with the tools necessary to attempt survival. Those that endure will grow slowly, passing through stages marked by incremental increases in size and capability. Muscles strengthen, neural networks refine, and behaviors become more precise with experience. Over time, the small translucent form may become an adult capable of navigating currents, locating mates, and repeating the cycle. For most, however, the early days define their fate. The ocean accepts them without distinction, and only a portion continue forward. In this quiet arithmetic of life, abundance at the beginning compensates for uncertainty along the way, ensuring that even within immense mortality, continuity persists. Along the seafloor surrounding the brooding grounds, life continues in forms that seldom draw attention. Small crustaceans move with deliberate steps across mineral surfaces, their antennae sweeping slowly as they search for edible traces. Nearby, slender fish glide close to the substrate, bodies angled slightly downward as they watch for drifting particles or minute animals disturbed by passing currents. Each creature follows its own quiet routine, linked to others through exchanges of energy that form an intricate web. Most nourishment here arrives indirectly. Organic material descends from distant waters above, transformed many times before reaching these depths. By the time it settles, it may exist as fragments of tissue, microscopic debris, or delicate films coating stone. Scavengers specialize in locating such remnants. Amphipods, worms, and other small invertebrates detect chemical hints and gather in modest numbers, feeding slowly and methodically. Their activity breaks larger particles into finer ones, making nutrients accessible to organisms that feed on still smaller scales. Fish contribute to this cycle as both hunters and participants. Some consume the crustaceans, while others sift through sediment for edible grains. Each feeding event redistributes matter, converting it from one form into another. Waste released by one species becomes nourishment for microbes or detritivores nearby. In this way, the food web resembles a chain of gentle transformations rather than a series of sudden exchanges. When a brooding female completes her vigil and her body grows still, the process continues without pause. Her tissues, once sustained by stored reserves, become a concentrated source of organic material. Chemical signals released during decomposition spread through the surrounding water, subtle yet detectable. Scavengers approach gradually, guided by these traces. They do not arrive in frenzy. Instead, they gather in small numbers, each taking part in the slow conversion of flesh back into circulating nutrients. Bacteria and other microorganisms begin the transformation first. Enzymes break complex molecules into simpler compounds, releasing elements such as nitrogen, phosphorus, and carbon into the immediate environment. These dissolved nutrients diffuse outward, where they may be absorbed by microscopic life or incorporated into mineral films coating nearby surfaces. Larger scavengers follow, consuming what remains and dispersing fragments across the seafloor. Nothing lingers unused for long. This recycling sustains the surrounding community. The nutrients derived from one organism pass through many others, entering new bodies and supporting new growth. A crustacean that feeds upon this material may later be eaten by a fish. That fish may in turn nourish another predator or release waste that enriches microbial colonies. The pathway extends outward through countless interactions, each step preserving energy within the system rather than allowing it to vanish. Even the smallest residues play a role. Trace minerals liberated during decomposition can bind to particles of sediment, altering the chemical composition of the substrate. Microbes living there adapt to these changes, adjusting their metabolism and influencing the availability of nutrients for other species. The entire environment responds in subtle increments, shaped by contributions from each organism that enters the cycle. In this realm, endings merge seamlessly with beginnings. What appears as conclusion for one life becomes continuation for many others. Matter shifts form, energy changes hands, and the web remains intact. Within such continuity, the deep ocean maintains its balance, sustained by a principle that governs every level of existence here. Nothing is discarded, and nothing is truly lost. In coastal waters where sunlight filters down in shifting bands, other octopus species follow a similar yet distinctly different path. There, reefs and rocky crevices provide sheltered chambers for brooding, and temperatures fluctuate with tides and seasons. A female may select a cave beneath coral or a narrow hollow in limestone, attaching her eggs to the ceiling of her chosen refuge. The setting feels dynamic, shaped by waves, currents, and changing light. In such environments, incubation unfolds more quickly. Warmer water accelerates metabolic processes within the developing embryos, shortening the span between laying and hatching. For many shallow species, this period may last only a few weeks or months rather than stretching across much longer intervals. The mother remains attentive throughout, cleaning and ventilating the clutch, yet the duration of her vigil reflects the higher ambient temperature that surrounds her. Shallow habitats also host a broader array of predators and competitors. Fish patrol reefs in constant motion, crustaceans hide within crevices, and other cephalopods share the same terrain. To counter these pressures, brooding females often rely on concealed dens and active camouflage. Their skin shifts rapidly in color and texture, blending with algae, sand, or coral patterns. Movement remains cautious, but not entirely restrained, as the dynamic surroundings demand alert responses to passing threats. In colder, deeper waters, the rhythm changes. Lower temperatures slow cellular activity, extending embryonic development and lengthening the brooding period. The mother's commitment, therefore, spans a greater fraction of her life. Energy must be conserved more rigorously, and the protective posture becomes nearly continuous. Rather than adjusting to waves or fluctuating light, she adapts to stability, maintaining position within an environment defined by constancy. The scale of aggregation also differs markedly. Most shallow water octopus species brood alone, separated from others by territories established through competition or simple spacing. Encounters between brooding females are uncommon, as suitable dens are dispersed across reefs and rocky coastlines. Each mother occupies her own chamber, isolated except for brief interactions during mating seasons. In contrast, the congregation along the seamount slope represents an unusual concentration. Hundreds or even thousands of females align along a narrow band of terrain, each attached to adjacent surfaces, yet maintaining individual broods. The proximity does not imply cooperation in the human sense, but it does reflect a shared selection of habitat. The conditions present there, particularly the subtle warmth and stable substrate, appear sufficient to attract many individuals simultaneously. This density creates a visual impression rarely seen among octopuses elsewhere. Instead of scattered dens separated by stretches of reef, the brooding bodies form a continuous presence across the rock face. Each female remains solitary in behavior, yet collectively they compose a living mosaic. The aggregation stands apart from typical patterns observed in coastal species, where environmental diversity disperses individuals across wider areas. Despite these contrasts, underlying principles remain consistent. Across depths and climates, females invest fully in their clutches, sacrificing feeding and mobility to safeguard development. Temperature shapes timing, habitat shapes spacing, and ecological pressures shape behavior. The differences reveal how flexible the octopus life strategy can be, adapting to varied surroundings while preserving its essential structure. Within this adaptability, the seamount gathering emerges not as an anomaly, but as one expression of a broader biological theme, intensified by the particular conditions of its setting. Far below the reach of storms and tides, the Earth sometimes releases its inner heat in dramatic fashion. In certain regions of the ocean floor, towering chimneys rise like dark spires, formed as superheated water surges upward from deep within the crust. These structures, often called black smokers, discharge mineral-rich fluid at temperatures hot enough to shimmer even in lightless depths. The water pouring from them carries dissolved metals and sulfides, which solidify upon contact with the surrounding sea, building columns that grow slowly year after year. Such vents create environments defined by intensity. Chemical plumes billow outward, dense with compounds that would be harmful to many forms of life. Yet specialized organisms thrive there, drawing energy not from sunlight, but from chemical reactions. Bacteria cluster in thick mats, crustaceans gather in dense swarms, and unusual animals adapted to these conditions populate the immediate area. The scene is active, shaped by constant discharge and rapid mineral deposition. The seafloor refuge, where the octopuses gather, differs profoundly from those volcanic outbursts. Here there are no chimneys, no clouds of darkened water, no torrents of heated fluid. Instead, warmth emerges gradually, filtered through layers of rock that soften its intensity long before it reaches the surface. The water that seeps upward carries only faint traces of dissolved minerals, not enough to stain the surrounding sea or alter it dramatically. The effect is gentle rather than forceful, subtle rather than spectacular. Temperature measurements reveal this distinction clearly. At hydrothermal vents, readings can climb to extremes that would damage delicate tissues. In this quieter setting, the warmth rises only slightly above the ambient baseline, forming narrow patches where conditions shift by just a few degrees. Such a difference might seem negligible in ordinary circumstances, yet in the deep ocean, even minor variations can influence biological processes. Organisms accustomed to stable cold respond noticeably to these modest increases. Chemical composition also remains comparatively mild. The seepage lacks the concentrated sulfides and metals characteristic of active vent systems. Without those substances, the surrounding water stays clear, and the seafloor retains its natural coloration. Microbial communities still develop, but they do so in thin layers rather than thick blooms. The entire environment feels restrained, shaped by slow exchange instead of vigorous release. This restraint is precisely what allows certain species to use the area without harm. Extreme vent conditions demand specialized adaptations that few organisms possess. The softer geothermal presence here creates a middle ground, offering warmth without toxicity, stability without turbulence. Such balance provides a setting where sensitive life stages can proceed undisturbed, supported by consistent conditions rather than challenged by volatile ones. The importance of this subtle heat lies in its reliability. Unlike vents that may flare or fade as geological activity shifts, low-temperature seepage can persist steadily for long spans of time. Rock acts as a buffer, moderating fluctuations and releasing warmth gradually. This continuity establishes a predictable environment, one that organisms can depend upon across generations. In the vast quiet of the deep, dramatic phenomena are rare and localized, while gentle processes extend quietly across wider regions. The distinction between these two expressions of geothermal energy illustrates how varied the seafloor can be. One place roars with mineral fire, another exhales a soft warmth through stone. Both originate from the same planetary forces, yet each shapes life in its own measured way. Observation in the deep ocean unfolds as a patient practice, guided by instruments that return again and again to the same coordinates. Researchers do not watch continuously with human eyes. Instead, they rely on machines that descend through the water column and hover with quiet precision above the seafloor. These remotely operated vehicles carry cameras, sensors, and measuring tools, each designed to record small changes that would otherwise pass unnoticed. When such a vehicle arrives at the brooding grounds, its lights reveal a scene already in progress. The cameras begin their steady recording, capturing wide views and close details alike. Video collected during one visit becomes a reference for the next. By comparing footage across months and years, scientists can detect shifts in arrangement, density, and condition. A cluster that appears unchanged at first glance may show subtle differences when examined frame by frame. An arm repositioned, an empty space where an individual once rested, or new eggs where none were seen before. Population counts emerge from these recordings. Through careful review, researchers slow the footage, marking each visible octopus and noting its position relative to surrounding rock features. The process requires concentration and repetition, since individuals often resemble one another closely. Digital overlays help track locations, allowing counts from different expeditions to be aligned and compared. Over time, these tallies reveal patterns of arrival and departure, showing whether numbers remain steady, increase, or diminish. Egg tracking follows a similar method. Cameras focus on selected clutches, returning to them during later visits to document development. Scientists examine the capsules for changes in size, clarity, and internal structure. By noting when embryos first become visible and when capsules appear empty, they estimate incubation duration. These observations accumulate into timelines that describe how long brooding typically lasts under local conditions. The timing of mortality can also be inferred through repeated surveys. An individual seen guarding eggs in one recording may appear motionless or absent in a later one. When such changes are documented across many observations, researchers can approximate the sequence that follows reproduction. They correlate these visual records with environmental measurements gathered by onboard instruments, including temperature and water chemistry. The combination of imagery and data provides context, linking biological events with surrounding conditions. Long-term monitoring depends on consistency. Vehicles are guided to the same mapped points using navigation systems that reference seafloor contours. Pilots adjust position carefully, aligning each new recording with earlier ones, so comparisons remain accurate. The result is a growing archive of visual and numerical information, extending across seasons and years. Each entry adds another layer of understanding, revealing trends that short visits could never capture. From this accumulation of evidence, reproductive cycles become clearer. Patterns emerge showing when brooding individuals are most numerous, when hatchlings appear, and how frequently new groups arrive. The process resembles assembling a mosaic from many small tiles. No single expedition provides the full picture, yet together they form a continuous record of life unfolding in that secluded place. Through such patient observation, the hidden rhythms of the site gradually come into view. Machines return, lights glow softly, and cameras watch without interruption. The ocean continues its quiet motions while the record grows, one measured descent at a time. Weight does not descend from above in the deep ocean. It surrounds, presses, and settles from every direction at once, an unseen force carried by the immense column of water overhead. At a depth of more than 3,000 meters, that pressure rises beyond 300 times what bodies experience at the surface. It is not felt as squeezing or crushing in the usual sense, because it acts evenly on all sides. Instead, it exists as a constant condition, a physical law that shapes every structure living within it. For human beings, such force demands protection. Submersibles built to visit these depths must be engineered with thick, reinforced hulls, often formed from titanium or specially treated steel. Their walls curve inward in smooth spheres or cylinders, shapes chosen because they distribute pressure evenly and prevent weak points. Windows are small and made from dense acrylic or quartz, designed to withstand the steady load of water bearing down upon them. Without such precautions, the surrounding force would collapse ordinary materials in moments, folding them inward with silent efficiency. Creatures native to these regions require no mechanical shield because their bodies have never known any other state. Over countless generations, evolution has shaped them to exist in equilibrium with the pressure around them. One of the most important adaptations lies in the absence of air-filled spaces. Gas compresses easily, shrinking as pressure increases, which would distort tissues and disrupt internal balance. Deep-sea animals therefore rely instead on fluids and soft solids, substances that resist compression and maintain their volume even when external force rises. Within their bodies, water-based tissues dominate. Cells are filled with liquid, organs are supported by gelatinous matrices, and skeletal elements are reduced or flexible. This composition allows pressure to pass through them rather than act against them. Instead of resisting the surrounding force, they match it, maintaining internal conditions that equalize with the environment. The result is stability. Structures remain intact, chemical reactions proceed normally, and movement continues without strain. Proteins inside these organisms also carry subtle modifications. At high pressure, molecular shapes can shift, altering how enzymes function. Deep adapted species possess biochemical variants that remain stable under such conditions, ensuring that essential reactions unfold correctly. Membranes surrounding their cells contain lipids arranged to preserve fluidity, preventing rigidity that might otherwise interfere with transport of nutrients and signals. These microscopic adjustments form an invisible architecture supporting life where physical laws might seem prohibitive. Blood chemistry reflects similar tuning. Oxygen must be transported efficiently despite the density of the surrounding medium. Specialized respiratory pigments bind and release gases in ways suited to pressure and temperature, maintaining steady supply to tissues. Circulatory systems operate at measured rates, calibrated to balance energy use with environmental demands. Nothing within these bodies is accidental. Each trait contributes to harmony with the immense force outside. For visiting humans enclosed in metal vessels, the pressure remains an adversary held at bay. For the animals who dwell there, it is neither enemy nor burden. It is simply the medium in which existence unfolds, as ordinary to them as air is to those at the surface. In that silent depth, strength is not defined by resistance, but by accommodation, by the quiet capacity to live within weight that never lifts. Darkness in the deep ocean is not simply the absence of light. It is a permanent condition, a surrounding element as constant as the water itself. Sunlight fades rapidly with depth, absorbed and scattered until no trace remains. Beyond that boundary, illumination never returns, not by day or by season. Life continues there without any expectation of brightness, shaped from its beginning to exist within unbroken night. Creatures that dwell in this realm do not depend on sight as surface animals do. Eyes may still be present, yet their role changes. Instead of forming detailed images, they register faint contrasts or subtle glimmers produced by living organisms. Vision becomes supplementary rather than central. Orientation arises from other senses, each attuned to signals that travel more reliably through darkened water. Chemoreception serves as one of the most important guides. Dissolved molecules drift through the sea, carrying information about nearby life, distant habitats, and recent events. Specialized receptors on skin and appendages detect these chemical traces, translating them into patterns the nervous system can interpret. A faint increase in a particular compound may indicate food, a potential mate, or a change in surroundings. Because chemicals disperse gradually, they create gradients that can be followed, leading an animal step by step toward their source. Touch provides another channel of perception. Many deep-dwelling species possess sensitive surfaces capable of detecting minute vibrations. Movements made by other organisms send ripples through the water, and these ripples travel far without interference. By sensing their direction and intensity, an animal can determine where activity is occurring even when no light reveals it. The seafloor itself also offers tactile information. Texture, slope, and firmness communicate position and orientation, forming a physical map that can be read through contact alone. Sound plays a subtle role as well. In water, vibrations propagate efficiently, carrying low-frequency signals across long distances. Some animals produce faint noises through movement or internal processes, and others are able to perceive them. These cues add another layer to environmental awareness, blending with chemical and tactile signals to create a complete sensory landscape. Navigation within this darkness depends on integrating all these impressions. Instead of relying on a single dominant sense, deep-sea organisms combine multiple inputs, each contributing part of the whole. The nervous system assembles these fragments into a coherent understanding of surroundings, allowing movement that appears purposeful and precise. Paths are traced not through sight lines but through gradients, currents, and textures. Without sunlight, Energy for ecosystems must originate elsewhere. Some regions rely on organic matter descending from distant waters, while others draw upon chemical reactions occurring within the seafloor. In either case, life adapts to conditions that remain stable for long periods. Rhythms are set not by day and night, but by internal cycles and environmental consistency. Activity may follow patterns of temperature, nutrient availability, or reproductive timing rather than the rising and setting of a sun that never reaches these depths. Within such an environment, perception becomes an intimate dialogue between organism and medium. Every movement, every chemical trace, every faint vibration carries meaning. Darkness does not conceal the world from those who live there. It simply defines the language through which the world is known. Energy governs every movement in the deep ocean, though it is rarely visible as motion. It exists instead as a quiet accounting, a balance measured within cells and tissues. Each organism carries a limited supply drawn from the sparse nourishment available in its surroundings. Because new resources arrive slowly, life here unfolds according to a careful budget where every action must justify its cost. Food does not appear in abundance. Nutrients drift downward in scattered fragments, and long intervals may pass between opportunities to feed. Animals adapted to this environment have evolved physiologies that stretch each intake as far as possible. Metabolic rates remain low, allowing stored energy to last for extended periods. Hearts beat steadily, but without urgency, and muscles contract only when necessary. This restraint is not lethargy. It is a strategy shaped by scarcity, ensuring that reserves endure through uncertain conditions. Growth reflects the same principle. In warmer regions, rapid metabolism supports swift development, yet in colder depths, biochemical reactions proceed more slowly. Cells divide at measured intervals, tissues form gradually, and maturity arrives after long spans. Some deep-dwelling species take years to reach full size, their bodies accumulating mass with patient consistency. The pace resembles geological time more than seasonal change, aligning life's rhythm with the unhurried character of its environment. Cold water contributes to longevity as well. Lower temperatures reduce molecular activity, slowing processes that lead to cellular wear. Proteins degrade more gradually, and metabolic byproducts accumulate at reduced rates. As a result, many inhabitants of the deep ocean live far longer than relatives in warmer habitats. Lifespans extend not because danger is absent, but because internal chemistry advances with deliberate steadiness. Within such conditions, reproduction represents the most significant expenditure an organism can undertake. Producing eggs or sperm requires substantial material, including proteins, lipids, and minerals that must be gathered from limited food sources. For a female, the cost increases further when brooding begins. Energy that once sustained her own tissues is redirected toward maintaining conditions suitable for developing offspring. The investment is total, leaving little reserve for recovery afterward. This pattern reveals the logic of deep-sea energy economics. When resources are scarce and growth is slow, opportunities to reproduce may be rare. It becomes advantageous to concentrate effort into a single decisive event rather than attempt multiple cycles that might never be completed. By allocating all remaining energy into one reproductive phase, an organism maximizes the likelihood that its genetic lineage will continue. The strategy favors certainty over repetition, completion over prolongation. Even behaviors reflect this accounting. Movement is minimized unless it leads to nourishment or mating. Encounters are brief, actions precise, and rest, occupies much of the time between. Each motion carries an energetic cost, and unnecessary expenditure could shorten survival. The result is a world where activity appears sparse, yet remains finely tuned to necessity. Across this quiet landscape, life persists through efficiency rather than abundance. Every cell operates within a narrow margin, balancing intake against use. Reproduction stands as the culmination of that balance, the moment when accumulated energy is released into the future. In the deep ocean, existence is sustained not by excess but by careful distribution, a steady arithmetic written into the living fabric of each creature that dwells there. Beneath the wide sweep of the open ocean, the seafloor is not uniformly flat. Mountains rise where tectonic forces once pushed molten rock upward, forming peaks that remain hidden beneath thousands of meters of water. These underwater elevations interrupt the steady flow of currents, altering movement in ways that ripple outward through entire ecosystems. Though unseen from above, their presence reshapes the surrounding sea with quiet persistence. When a current encounters the slope of a seamount, it cannot pass straight through. Water is guided upward along the incline, a process known as upwelling. As deeper layers ascend, they carry with them dissolved nutrients that have accumulated over long periods. These nutrients often include nitrogen compounds and phosphates derived from decomposed organic matter. Lifted toward shallower depths, they become available to microscopic organisms that form the base of marine food webs. This vertical mixing enriches the water column around the mountain. Phytoplankton in regions where light still penetrates may increase in abundance, drawing energy from sunlight and incorporating the newly available nutrients into their tissues. Even at greater depths, suspended particles and microbial communities benefit from the redistribution. The seamount acts as a subtle pump, circulating elements that would otherwise remain dispersed or trapped in lower layers. Currents interacting with these elevations also create eddies and localized flow patterns. Such movements can concentrate drifting organisms, bringing prey and predator into closer proximity. Larval stages of many species may become retained near the mountain rather than swept away into open ocean. Over time, this retention supports stable populations that persist in association with the structure. Hard substrate provided by volcanic rock offers another essential resource. In vast stretches of deep ocean, soft sediment dominates the seafloor, limiting attachment opportunities for organisms that require firm surfaces. Seamounts present cliffs, ridges, and exposed rock faces where corals, sponges, and other sessile animals can anchor themselves. These communities form intricate habitats, adding three-dimensional complexity that shelters numerous smaller species. Because of these combined effects, seamounts often function as biodiversity hotspots. Species richness tends to exceed that of surrounding plains, and unique assemblages may develop in relative isolation. Fish congregate to feed along nutrient-enriched currents, while invertebrates colonize every available niche. The mountain becomes a focal point, a gathering place shaped not by deliberate design, but by physical interaction between geology and water. Migration routes may also intersect with these underwater landmarks. Animals traveling across open expanses can use them as orientation points or feeding stations. In this way, seamounts contribute to broader connectivity within the ocean, linking distant regions through shared use of localized resources. The influence of a single elevation can therefore extend far beyond its immediate footprint. The attraction of life to these hidden mountains arises from balance rather than abundance alone. Upwelling supplies, nutrients, rock provides structure, and altered currents create stability within motion. Together, these factors establish conditions favorable for persistence and diversity. The result is a living community woven into the contours of stone, sustained by forces that operate quietly and continuously. Within the immense blue expanse, seamounts stand as reminders that topography shapes biology even where no human eye can see it. Water flows, nutrients circulate, and organisms respond. Life gathers not by chance, but through the steady interplay of earth and ocean, meeting along slopes that rise unseen beneath the waves. Long before instruments could descend into the ocean's depths, human understanding of the deep sea existed only in imagination. Philosophers once reasoned that life could not persist so far below the surface, believing that darkness, pressure, and cold would render those regions barren. The abyss was described as an empty realm, a place beyond the reach of vitality. For centuries, this assumption endured, supported not by evidence but by absence of observation. Gradually, curiosity pressed downward. In the 19th century, a British research vessel set out to examine the oceans with systematic intent. The voyage of HMS Challenger marked a turning point in scientific exploration. Between 1872 and 1876, its crew collected samples from previously unreachable depths, lowering weighted lines and dredges into waters thousands of meters deep. When these instruments returned, they carried mud, minerals, and living organisms, delicate creatures that contradicted long-held expectations. The findings revealed that life existed even in the most remote regions of the sea, quiet yet unmistakable. This expedition did more than gather specimens. It established methods for studying the ocean as a global system. Measurements of temperature, salinity, and depth were recorded with care, creating one of the first comprehensive datasets about the marine environment. Charts produced from these observations transformed blank spaces on maps into documented terrain. Though much remained unknown, the notion of a lifeless deep began to fade. In the decades that followed, technological progress opened new pathways into the depths. Early submersibles allowed brief descents, their crews peering through small viewports at landscapes no human had witnessed directly. Later designs improved endurance and maneuverability, enabling longer visits and more detailed observation. Remotely operated vehicles extended this reach further, transmitting images to the surface while enduring pressures no human body could tolerate. Through these tools, the seafloor gradually revealed its contours and inhabitants. Each advance brought fresh discoveries. Mountain ranges hidden beneath water were charted, trenches of immense depth were measured, and unfamiliar species were documented. Yet the scale of the ocean ensured that progress remained slow. Mapping the seafloor requires systematic surveying, often using sonar pulses that travel downward, reflect from the bottom, and return with information about shape and distance. Even with modern instruments, covering vast areas demands time and repeated passes. Because of this challenge, a surprisingly small portion of the ocean floor has been mapped in high resolution. Large expanses remain known only through coarse measurements, their fine details still concealed. Compared with the surfaces of nearby planets, which have been charted extensively by orbiting spacecraft, Earth's own seabed is less completely documented. The reason lies not in lack of interest, but in practical difficulty. Water absorbs and scatters signals, and great depth complicates direct observation. Despite these limitations, exploration continues with steady determination. Research vessels trace survey lines across remote regions, and autonomous vehicles glide through the water collecting data. Each mission adds another fragment to the growing picture of the deep. Knowledge accumulates gradually, like sediment settling layer by layer. From early speculation to modern investigation, the history of deep-sea exploration reflects a shift from assumption to evidence. What was once imagined as empty is now recognized as intricate and inhabited. Yet much remains to be seen. Beneath the ocean's surface lies a realm still largely uncharted, waiting in silence while instruments descend and return, carrying glimpses of what endures below. Discovery sometimes arrives not as a shout but as a quiet correction. For generations, marine biology described octopuses as solitary creatures whose lives unfolded in isolation, each individual occupying its own small territory. Observations from shallower environments supported this belief, and without contrary evidence from great depth, the idea settled into scientific understanding like sediment. The gathering along the volcanic slope altered that perception, not abruptly, but with the steady authority of direct observation. What researchers documented there was unprecedented in scale. Instead of scattered individuals separated by distance, hundreds and then thousands appeared within the same confined area, each engaged in the same reproductive phase. The pattern suggested coordination without communication, a shared response to environmental conditions rather than social behavior. Such density had never been recorded for deep-dwelling octopuses, and its existence required scientists to reconsider assumptions about how these animals locate suitable breeding grounds. The site revealed that habitat selection could be far more deliberate than previously recognized. Subtle variations in temperature, chemistry, and substrate formed a combination precise enough to attract large numbers of females at the same stage of life. This indicated that individuals were not arriving randomly. They were detecting specific cues and converging upon them, demonstrating an ability to evaluate their surroundings with refined sensitivity. The implication extended beyond a single species, suggesting that other deep-sea organisms might also respond to environmental gradients in ways not yet observed. Equally important was the window this aggregation opened onto reproductive behavior. Until such gatherings were recorded, much of what scientists understood about octopus breeding came from species living in accessible waters. The deep ocean remained largely speculative, its inhabitants seldom witnessed during critical life stages. Here, for the first time, researchers could follow a population through the full progression of brooding, from arrival to development to the final stillness that followed. This continuity provided a timeline grounded in evidence rather than inference. The discovery also prompted reevaluation of life history strategies among cephalopods as a whole. If one species could concentrate in such numbers under the right conditions, it raised the possibility that similar events might occur elsewhere, hidden along other remote slopes or ridges. Biological patterns once thought rare or exceptional might in fact be widespread, simply overlooked because so little of the seafloor has been examined closely. Each new observation therefore carried broader relevance, informing theories about growth, survival, and reproduction across related species. Scientific understanding rarely shifts through speculation alone. It changes when observation reveals something the existing framework cannot easily explain. The congregation on that distant incline provided exactly such evidence. It showed that even well-studied groups can hold behaviors unknown to science, waiting in places seldom visited. With that realization came a quiet adjustment in perspective. The deep ocean was not only inhabited, it was behaviorally complex, shaped by interactions subtle enough to remain concealed until technology and patience aligned. In this way, the site became more than a biological curiosity. It became a reference point, a reminder that knowledge of the natural world remains incomplete, especially in regions difficult to reach. Each recorded image, each measured detail, contributed to a revised understanding of how life persists and reproduces far below the surface. What once seemed improbable now stood documented, inviting science to expand its sense of what is possible. Quietly, knowledge deepens as exploration continues, and the hidden slopes of the world yield their truths one observation at a time, reminding those who study them that discovery is not a single moment, but a long unfolding, patient as the ocean itself. It endures in silence still. Far beneath the surface, change moves slowly. Yet even the most gradual shift can alter conditions that once seemed constant. The deep ocean has long been defined by steadiness, its temperatures varying only slightly across centuries. This stability has shaped the biology of its inhabitants, whose bodies and life cycles are tuned to narrow environmental ranges. When those ranges begin to move, even subtly, the effects can extend through entire communities. One influence arises from the warming of surface waters. Heat absorbed at the ocean's upper layers does not remain there indefinitely. Over time, currents carry portions of that warmth downward, distributing it into deeper strata. The process unfolds gradually, often measured in fractions of a degree. Yet organisms accustomed to nearly unchanging temperatures may respond to these increments. Physiological reactions, metabolic timing, and developmental rates all depend on thermal conditions, and small variations can alter their balance. Another shift occurs through the absorption of carbon dioxide from the atmosphere. When this gas dissolves into seawater, it forms carbonic acid, lowering pH levels in a process known as acidification. Though the change is slight, it influences chemical interactions throughout the marine environment. Minerals used by certain organisms to build shells or skeletal elements become less available, and biochemical pathways within cells may function differently. In deep regions, where circulation is slow, such chemical adjustments can persist for long intervals before dispersing. Habitats shaped by geothermal activity may face particular sensitivity. Their physical structure depends on delicate exchanges between heated fluids and surrounding seawater. Alterations in temperature or chemistry could modify how these exchanges occur, potentially shifting the boundaries of suitable habitat. Because such environments often occupy limited areas, any transformation may affect the species that rely upon them with disproportionate intensity. Reproductive sites require especially consistent conditions. Developmental processes within eggs proceed according to precise environmental cues, including temperature, oxygen concentration, and chemical composition. If these factors drift beyond accustomed ranges, timing may change, growth may slow, or survival may decline. For species whose reproductive strategy depends on a single major effort, stability becomes not merely beneficial, but essential. Their life histories assume that the environment will remain within predictable limits long enough for offspring to mature. The deep ocean's reputation for constancy once suggested that it might remain insulated from surface-driven climate shifts. Ongoing observations indicate that this assumption may not hold entirely true. Signals originating above can propagate downward through circulation, mixing, and gradual diffusion. The pace remains slow compared with atmospheric change, yet it is steady, and the cumulative effect over decades can become measurable. Scientific monitoring continues to examine these patterns, comparing present measurements with historical records to detect trends. Instruments anchored to the seafloor or carried by research vehicles track temperature, chemistry, and current behavior across extended periods. Each dataset adds detail to an emerging picture of how deep environments respond to planetary scale influences. Within this unfolding context, stability reveals itself as a quiet foundation supporting life far below. Where conditions remain steady, biological rhythms proceed with assurance. Where they shift, even slightly, organisms must adjust or yield. The balance is delicate, maintained through interactions that operate without haste, yet never entirely at rest. Nothing in the deep ocean is discarded. What appears to end does not vanish but changes form, passing quietly from one existence into another. In this vast environment, where resources arrive sparingly and every fragment carries value, the boundary between life and death softens into a gradual transition. Matter is never idle for long. It drifts, settles, dissolves, and is taken up again, entering new bodies and new processes without interruption. When an organism's life concludes, its tissues begin a gentle transformation. Microbes present in the water and sediment respond first, breaking complex molecules into simpler compounds. Their activity releases nutrients that had been bound within living cells, returning them to the surrounding environment. These microscopic decomposers operate steadily, their work invisible yet essential, initiating a chain of exchange that sustains other forms of life. Scavengers soon participate as well. Small crustaceans, worms, and fish detect chemical traces that signal the presence of organic material. Guided by these cues, they arrive and feed, incorporating portions of that material into their own bodies. What they consume becomes energy for movement, growth, and reproduction. What remains continues to fragment, passing downward through successive stages of breakdown. Each participant takes only what it requires, leaving the rest for others. Sediments beneath these exchanges serve as reservoirs, collecting fine particles that settle from above. Within these layers, bacteria and archaea continue the process of decomposition, transforming residual matter through biochemical pathways. Minerals released during this process dissolve into surrounding water, where currents can transport them to distant locations. In time, they may nourish organisms far from their origin, illustrating how material circulates across space as well as through living systems. Because conditions in the deep ocean remain stable and cold, these transformations proceed slowly. Decomposition does not rush. Instead, it unfolds with measured continuity, allowing nutrients to persist and circulate over extended periods. This deliberate pace supports ecosystems adapted to scarcity, ensuring that resources remain available rather than being consumed all at once. The rhythm resembles a long breath, drawn out across years instead of moments. Within such a system, nothing is wasted. Every element that once formed muscle, skin, or shell eventually contributes to another structure or process. Carbon becomes part of microbial cells. Nitrogen enters dissolved compounds. Calcium joins mineral grains. Each substance follows pathways governed by chemistry and biology, together, guided not by intention but by natural law. The result is a seamless cycle in which endings nourish beginnings. These exchanges also connect generations. Material that sustained one individual may later support another, linking lives separated by time. In this way, continuity extends beyond single lifespans, weaving organisms into a shared fabric of matter and energy. The cycle persists regardless of scale, from microscopic communities to larger animals, each participating in the same enduring pattern. Far below the surface, this process continues without spectacle. There are no sudden changes, no abrupt transitions, only steady transformation. Life yields to death, death yields to nourishment, and nourishment yields again to life. The sequence does not repeat in identical form, yet it never ceases. It flows onward, carried by currents and chemistry, sustained by countless unseen interactions. Through this quiet circulation, the deep ocean maintains balance. Resources remain in motion, ecosystems remain supported, and existence continues its patient course. Nothing is lost. Everything returns, reshaped and renewed, within the vast and unending exchange that defines the living sea. Within the quiet chambers of an octopus body lies a nervous system of remarkable intricacy. Its neurons number in the hundreds of millions, rivaling those of many vertebrates, yet they are arranged in a pattern unlike our own. Rather than concentrating entirely within a single brain, much of this neural tissue extends outward into the arms, forming networks capable of processing sensation and directing movement independently. Thought, in such a creature, is not confined to one center, but distributed throughout a living architecture of touch and response. Each arm contains clusters of neurons that can evaluate texture, resistance, and chemical traces in the water. When an arm explores a surface, it does not wait passively for instructions from the central brain. It interprets information locally, adjusting grip and movement with subtle autonomy. The result is a fluid coordination between central command and peripheral intelligence, a partnership that allows rapid adaptation to complex surroundings. This arrangement gives the impression of awareness present in every reaching limb. Experiments conducted in controlled environments have shown that octopuses can learn from experience. They solve simple puzzles, remember pathways, and modify behavior after repeated trials. Such findings reveal cognitive flexibility, suggesting that these animals form internal representations of their environment. Learning, in this context, becomes part of survival, allowing individuals to refine hunting strategies or avoid potential threats. Yet alongside this capacity for learning operates another powerful influence. Deep within the body, endocrine signals guide development and reproduction according to internal timing. Hormones released at specific stages trigger changes in appetite, activity, and physiology. These signals do not arise from conscious deliberation, but from biochemical processes shaped by evolutionary history. When reproductive maturity is reached, cascades of hormones initiate behaviors and transformations that follow a consistent sequence. The coexistence of intelligence and hormonal programming creates a delicate balance. An octopus may demonstrate problem-solving ability, yet certain life events proceed independently of choice. During the reproductive phase, physiological shifts alter feeding patterns and metabolic priorities. Neural circuits respond to endocrine cues, directing behavior toward brooding and away from foraging. The animal's capacity to learn remains, but its biological priorities are reoriented by internal chemistry. In this interplay, reproductive death does not represent a decision in the human sense. It is not selected through reflection or preference. Instead, it unfolds as part of a life history strategy encoded within genes and expressed through hormones. The optic gland releases substances that influence the rest of the body, coordinating a progression from reproduction to decline. Neural complexity does not override this program. It operates within it. Understanding this distinction clarifies the nature of octopus intelligence. Cognitive sophistication allows flexible interaction with the environment, yet it exists alongside mechanisms that ensure the continuation of the species. Instinct and learning intertwine rather than compete. One guides immediate problem-solving, the other governs long-term biological cycles. The distributed nervous system contributes to this harmony. Because neural processing occurs both centrally and peripherally, The animal can maintain detailed sensory engagement even as hormonal shifts redirect broader patterns of behavior. Arms continue to explore and adjust, while overarching physiological changes proceed according to internal design. The result is a life shaped by both awareness and inevitability. In the deep ocean, where conditions demand efficiency and precision, such integration proves effective. Intelligence enables adaptation to immediate challenges, while instinct secures the generational rhythm. Neither element stands alone. Together, they form a coherent system, capable of complexity yet bound to cycles that extend beyond individual experience. Through this union of neural richness and programmed destiny, the octopus navigates its world, acting with responsiveness while following a path inscribed within its own living chemistry. Knowledge of the deep ocean advances in small, illuminated circles, each expedition casting light upon a limited area while immense distances remain unseen. What has been revealed offers clarity, yet it also outlines the boundaries of understanding. Beyond those boundaries lie questions that persist, not from lack of curiosity, but from the sheer scale and inaccessibility of the environment. One mystery concerns how individuals locate the same remote breeding grounds with such consistency. Subtle gradients in temperature or chemistry may guide them, or faint traces carried by currents might form invisible pathways through the water. It is possible that seafloor contours influence their movement, providing tactile routes along slopes and ridges. Each explanation holds merit, yet none has been confirmed in full detail. The signals involved may be so delicate that they escape present instruments, detectable only by senses evolved specifically for that realm. Uncertainty also surrounds long-term population stability. Observations gathered over years show patterns, though the span of human study remains brief compared with the timescales of deep ocean life. Populations might fluctuate slowly, responding to environmental changes that unfold across decades. Alternatively, they may remain steady for centuries, sustained by conditions that vary little from one generation to the next. Without records extending far into the past, these possibilities remain open. The timing of gatherings presents another unknown. Some aggregations may form regularly, tied to cycles within the environment, while others might occur only under particular circumstances. Shifts in currents, variations in chemistry, or geological processes beneath the seafloor could influence when and where such events arise. Because only a few sites have been documented carefully, scientists cannot yet determine how often these congregations appear, or whether they migrate gradually from one location to another. It is also likely that similar places exist beyond those already observed. The ocean floor spans vast regions that have never been examined closely. Hidden slopes and ridges may host communities comparable to those known today, their presence unrecorded simply because no instrument has passed above them. Each unexplored area carries the possibility of discovery, not as a distant hope, but as a statistical certainty given the immensity of the seafloor. Technological limitations contribute to this uncertainty. Even with advanced vehicles and sensors, surveying great depths demands time, resources, and favorable conditions. Mapping a single region in detail can require multiple expeditions, and the global ocean contains countless such regions. Progress, therefore, unfolds gradually, expanding knowledge piece by piece rather than all at once. These unknowns do not diminish what has already been learned. Instead, they frame it within a broader perspective, reminding observers that understanding is always partial. Every confirmed fact rests beside unanswered questions, each discovery accompanied by further possibilities. The deep ocean remains a place where explanation and mystery coexist, neither replacing the other. Within that balance, inquiry continues. Instruments descend, measurements accumulate, and interpretations evolve as new evidence appears. Yet even as knowledge grows, the larger expanse persists beyond reach, vast and intricate. The living systems of the deep sea carry on in quiet continuity, their full complexity still unfolding beyond the limits of present sight, waiting without urgency while human understanding slowly follows after. Above the ocean, dawn arrives with color and sound. Yet far below, another world continues without sunrise or horizon. It moves at a pace so unhurried that change seems almost invisible, though it never truly stops. Currents glide along unseen contours, organisms follow inherited patterns, and geological warmth rises through stone as it has for ages beyond memory. This hidden realm does not compete with the surface for attention. It simply persists, complete within itself. The places that remain out of sight often hold the most unfamiliar forms of life. Creatures there follow strategies shaped by conditions unlike those found anywhere else. Growth may take years, where it would take weeks in shallower seas. Movement may be measured and deliberate, conserving strength for moments of need. Reproduction may occur only once, timed precisely to environmental signals that arrive without fanfare. These patterns can seem improbable when imagined from above, yet they unfold with quiet reliability in their own setting. What appears unusual from one perspective may be ordinary from another. In that distant garden along the seafloor, existence follows a design that is neither grim nor wasteful. Each stage serves a purpose within a larger continuity. Arrival, brooding, and stillness form parts of a sequence refined through countless generations. Nothing in that progression is hurried or undone. It proceeds with the calm assurance of processes that have endured for ages. The deep ocean itself contributes to this sense of continuity. Its waters circulate slowly, guided by differences in temperature and salinity that shape global currents. These movements carry nutrients, distribute heat, and connect distant regions through gradual exchange. While storms reshape coastlines above, these deeper motions remain steady, tracing pathways that may take centuries to complete a single circuit. Life within them adapts to their rhythm, aligning biological cycles with environmental constancy. Far overhead, daily life continues. For those who dwell on land, cities brighten and darken, conversations begin and end, and time is measured in hours. Beneath that familiar pattern, thousands of meters down, another chronology unfolds. There, time is counted in growth rings, chemical gradients, and the slow maturation of living forms. The difference is not a contrast of importance but of scale, two tempos existing at once within the same planet. The existence of such a place invites reflection on how much of Earth remains beyond ordinary awareness. Vast regions lie hidden not because they are unreachable forever, but because they are distant from daily experience. Exploration offers glimpses, brief illuminations that reveal complexity where emptiness was once assumed. Each glimpse adds to understanding, yet the greater expanse remains, patient and undisturbed. Seen from this perspective, the deep sea is not a remote curiosity, but a continuing presence. Its processes influence global systems, its inhabitants participate in cycles that sustain the planet, and its landscapes shape currents that travel far beyond their origin. The garden below is one expression of that larger whole, a small yet intricate chapter within an immense story. As night passes over the surface and morning returns again, the depths continue in their steady course. Nothing there depends on sunlight to begin or end its day. Life proceeds according to rhythms older than tides, guided by forces that operate without noise or spectacle. In that quiet persistence lies a reminder that the world is broader than what is easily seen, and that far below, the ancient motion of living systems carries on, serene and unbroken, through every passing hour.